Our purpose is to share with you pertinent information from the forensic science community concerning the role of the crime scene investigator in identifying, recording, collecting, preserving, and transporting items present at the crime scene that could possibly produce DNA results that are meaningful or probative to the case. Each crime scene presents unique evidentiary challenges, and the crime scene investigator must be thoroughly prepared to handle each in a manner that will not compromise the integrity of the items of interest. This is particularly true of items that could possibly contain DNA, whether it is visible, biological, body fluids, materials such as blood, semen, hair, body tissue, bone, saliva, urine, or the unseen touch DNA material which may be on the hat of a sweatband, the shirt cuffs or its collar, or the fingerprint powder residue. You are most likely familiar with seeing biological stains at the crime scene, and in many cases have presumptively tested or had your lab provide a confirmatory test which indicates and identifies the presence of a particular type of body fluid, and then the stain was presented for DNA analysis. Touch DNA presents a new challenge, so let's take a moment to define what we mean by touch DNA and its ramifications. Epithelial or touch DNA is evidence with no visible staining that would likely contain DNA resulting from the transfer of epithelial cells from the skin to the object. It is reported that 6 to 10 cells can produce a full DNA profile. But keep in mind that even if the surface is touched and cells left behind, there is no guarantee of a meaningful DNA profile. Detecting and attaining an interpretable DNA profile are two different concepts. Today, we will concentrate on the detection and collection of that possible DNA evidence. Thus, the investigator's job is to find those objects that could possibly contain touch DNA. Ironically, our human structure makes it likely that a perpetrator and/or the victim may leave touch DNA on an item. The average person sheds approximately 400,000 cells daily, and it is highly probable that the perpetrator may touch his face or head and transfer a large number of the cells to his hands or his gloves and hopefully to the items he touches. Strong contact pressure applied increases the probability. Thus, scuffling or grabbing of clothing while committing the crime can help produce the evidence that we need. Aware of this probability and the fact that enhanced DNA testing can process as few as 6 to 10 skin cells to provide useful DNA information for the case, and DNA typing kits have been so improved sensitivity and the number of loci detected, and instrumentation has improved in the areas of extraction, quantitation, and instrument analysis, we can't afford to overlook this type of evidence. Accordingly, it is important that the investigator understand the basic principles of DNA analytical procedures and appreciates the significance that he or she plays in that role. In the next series of slides, we will identify some basic lab processing of the sample, the names and types of DNA analysis utilized, the impact of improved processing, and the probability of the results. As you can see in the first slide, the basic lab sequence involves sequential steps. The first is the extraction. This is when the DNA is removed or isolated from other cellular material in an evidence sample before testing. Next, we measure the amount of quantitative DNA that is present. Do we have enough DNA to conduct our testing? The next step involves the amplification or multiplication of copies of specific small regions so that a sufficient amount of material will be available for analysis. Finally, the resulting PCA procedures are separated and measured so that they they can be detected for characterization. And finally, an analysis and an interpretation is produced. It's important that we understand some of the terms that are used in the type of DNA analysis. In amplification, the first process that we are using is called polymerase chain reaction. This is a process in which the DNA is reproduced to millions of copies. This is especially useful in the highly degraded evidence, but it's a double-edged sword because the sensitivity makes PCR extremely susceptible to contamination issue, an issue that we must be fully concerned about as being crime scene investigators. The short tandem repeats refers to those specific reagents that have been identified as the identified quality for DNA evaluation. The variable nature of the SDR regions intensifies the discrimination between one DNA profile and another. We are also be able to identify the gender identification by looking at the ameliogen locus. This is the area that we can identify male and female. I also want to mention briefly mitochondrial DNA, a type of DNA that is found in the mitochondria of each cell. This type of DNA is used in archaeological studies or in military scenarios. It's interesting that the DNA of all human beings is actually nearly identical. Approximately 99.9% of the sequence of the base of DNA is in exact order. However, there are places on the human DNA that are different, and it is those places, the locuses or the loci, that we look at for forensic analysis. The FBI has chosen 13 specific non-coding. By non-coding, we mean that there is no known reason or function for that specific loci for the standard for CODIS. This ensures that all U.S. forensic science labs can establish uniform DNA databases and share this information with the rest of the forensic community. The next slide is an interesting slide in which we are trying to illustrate the improvements that have been made in DNA analysis. Up until the early 2000s, the process that was used for identifying or producing a DNA profile was called restriction fragment length polymerase. This was a very exacting process, but it required a significant amount of source material to conduct the test. And when we say significant amount, we're talking about 50 microliters, which is 1/1,000,000th of a liter. Today, using PCR and short tandem repeats, the standard is about 300 picoliters. The picoliters is 1/1000,000th of a liter. So the illustration that we have on the slide, the quarter size for the restricted fragment length process, as opposed to the question mark in the middle of the block for the PCR short tandem repeat, gives you an indication of how small and how significant the process would be in qualitating and examining small amounts of evidence. Now, let's get to what it's all about. How closely can our evidentiary sample be associated with a known sample? This is the exclusion and inclusion process that we go through. And inclusion is the ability to exclude DNA typing at a specific location in a DNA profile from a known individual. What we're hoping to do is we produce a full profile, the genetic makeup of an individual at defined locations. Typically, this consists of one or more alleles, different forms of genes or genetic markers at a minimum of 13 locations, plus the ameliogen locus. A match occurs when the sample allele at that locus matches the reference sample. We exclude when any one of those fails to match. We include when we cannot exclude a potential contributor from the evidentiary item based on the comparison of known and questioned DNA profiles. Stated probabilities, that is, the chance of a random, non-related individual, excluding identical twins, in the same ethnic group having the same identical STRs at target regions run from the probability of, if we had 3 short tandem matches, 1 in 5,000, to where we have 13 short tandem repeat matches to 1 in a trillion. Although these statistics are subject to challenge, the magnitude of the ratios makes DNA a most valuable comparative analysis process in forensic science. Realizing the analytical capabilities of our forensic science laboratories, it becomes obvious that the identification, collection, preservation, and transportation of those items has a great impact on what we do. Because DNA evidence is more sensitive than other types of evidence, law enforcement personnel must be especially aware of their actions at the scene to prevent inadvertent contamination of that evidence. And no matter how repetitious it becomes, we must review and adhere to the common rules that help minimize contamination and degradation of the biological evidence found at the scene of the crime. Let's review some of these rules. As you can see, common sense is the common rule behind all of these. Limiting access to the crime scene makes perfect sense. The fewer people we have in the crime scene, the less the possibility of contamination. Wearing personal protective equipment, including masks, shoe covers, coveralls, and hair covers, is a must. That helps us avoid talking, coughing, sneezing, perspiring over that evidence and contaminating it. Also, we must learn to change our gloves frequently when we handle evidence. We're talking about touch DNA evidence. So when we collect an area, there's a great chance that our gloves will come in contact with this surface. If they do, that touch DNA can easily transfer to those gloves, and if you use the same gloves at another site, be transferred to that site. So gloves should be changed after every collection. Obviously, we do not want to touch areas on the evidence that may contain the DNA. So when we're handling shirt or clothing or a hat found at the scene of the crime, we have to be careful not to touch the sweatband or the bill of that object, or the areas where we think that the DNA would be. It is important to consider using DNA-free or disposable fingerprint brushes, fingerprint powders, lifting tapes, and swabs. Also, any materials or any tools that are taken into the crime scene must be cleaned after their use. The common cleaning solution is a 10-to-1 solution of water and bleach, and we should make sure that we thoroughly dry that equipment before it's used again. Does it make sense not to touch the outside of the gloves to our face or hands, or use personal items such as cell phones and radios? Those are all items that could contaminate the precautions that we have taken, and that you have to resist the urge to answer your cell phone while you're in the crime scene. Collecting and eliminating samples from those who have been in contact with the evidence is mandatory. Just as we collected fingerprint samples to eliminate possibility of other people involved in the crime scene, we have to be prepared to take sample eliminations for DNA purposes. And naturally, we don't want to discard trash in or around the crime scene. Once thoroughly prepared to protect the integrity of the crime scene and safeguard and preserve the evidence found therein, we can proceed with our mission, keeping in mind that a few cells can be sufficient to obtain useful DNA information. The following list identifies some common items of evidence that you may need to collect, the possible location of the DNA on that evidence, and the biological source containing the DNA evidence. Note that this is a general guideline of evidence. It's not meant to be an exclusive list, and I won't take your time to go down each one of the areas. However, one of the things that always draws my attention is the common toothpick. How small an object could easily be overlooked. However, the tip of that toothpick and its shaft can contain very valuable evidence. We put it in our mouth, we touch it with our hands, and in that situation, our saliva or our epithelial cells could be on it. Also, I want to point out that in the source of DNA, we mentioned sweat. DNA is generally not contained in water. However, the sweat is carrying epithelial cells, which becomes the source of the material that we are looking for. We must keep in mind that just because we cannot see a stain does not mean that there are not enough cells for DNA typing. And DNA does more than just identify the source of the sample. Remember that DNA can place an unknown individual at a crime scene. It may place that individual in a home or a room where that suspect claimed not to be. It may refute a claim of self-defense and put a weapon in the suspect's hands, or it can change a story from an alibi to one of consent. The more the investigator knows how to use DNA, the more powerful the tool it becomes. Biological evidence and touch DNA material should be collected and packaged in a manner that prevents contamination and degradation and ensures integrity during all phases of the investigation and the litigation. The following clean collection practices should be considered. When field testing evidence, swab the stain and test the swab rather than directly testing the stain. If the stain is small, it should be tested in the lab rather than the field. When it is necessary to dampen a swab to collect dried stains, any source of water that does not contain human DNA is acceptable. Sterile water, distilled water, and saline solutions meet this definition. The investigator must document which type of water he has used. The following videos cover the types of collection devices that can be used to collect biological and/or touch evidence, and the second one will illustrate a collection process.
As we talk about DNA collection, we can talk about various types of collection devices. We want to briefly go over some collection devices that are available in the market. The first device is a simple cotton swab with a wooden shaft. The challenges of this item is that the wooden shaft can help to collect bacteria, and in addition, because of the wood and the cotton combination, this swab will take the longest to dry. The next level, again, is a cotton tip swab, but this one has a synthetic shaft that allows it to dry faster but still collect the same level as the basic cotton swab. The next level eliminates having to do the pre-drying before putting it into a container. This is a swab, a cotton swab with synthetic shaft that is contained in its own protective sleeve. Once the swab is taken, it can be placed back into the sleeve and protected from any outside contaminants. Because of the membrane, it allows it to dry. One of its challenges is that it can take a long time to dry in a very humid atmosphere. The highest level of swab is, again, a cotton swab with synthetic shaft, but in its own container. But inside this container, once the swab is placed back in, there is a desiccant. This desiccant allows this swab to continue to dry regardless of the outside atmosphere. In addition to these swabs, there are some other specialty swabs that are also available. One of them, the Omni or SEP swab, is made with a compressed paper at the end. It has a unique shape which allows it to be more effective in collecting on certain surfaces. Relatively new to the market is a flocked swab. It's about the same area as the cotton swab on the head, but this is made of a loose tanglement of nylon fibers. It collects about the same level of DNA from the surface, but studies have shown that in the laboratory it has a higher release rate of DNA material. This swab is used for the wet and dry method. It has been shown that a wet swab and a dry swab for touch DNA on certain surfaces is more effective. This allows you to take the wet and the dry sample, place them both back into the container, and then because of the desiccant, allow them to dry. Let's return to the cotton swab. If we take a sample with a cotton swab, we have to make sure that this swab is absolutely dried before turned into evidence. The simplest form is to place the swab into a dry box and allow it to dry in the atmosphere. One of the challenges is that it is still susceptible to contamination by doing this. We could also air dry and place this in something called a capture device. This is where the swab is taken out, the swab is taken, and then can be pulled back into this and sealed, allowing it to dry, but the challenge is that it still is open to contamination from the air that enters it. A swab that is dried can be placed into a swab box. This can be sealed and labeled to enter into evidence. The challenge of this box is that if it's not completely dry, it is touching the edges and could be affected. More complex box, which eliminates this problem, would be this swab box, which actually has a container that holds the swab without touching the edges or surface. This can be closed with the swab inside and then allowed to dry and transported for evidence, protected from the contaminants in the atmosphere and also not touching the sides of the box. Finally, when it comes to swabbing, we want to make sure that we're using something besides tap water. We want to use a sterile water, whether it be in a 3 milliliter container like this or with a drop bottle. We only need to moisten the surface of the swab during sampling, but we want to make sure that we do that with some sort of sterile water at the scene.
In a previous video, we discussed the different types of collection devices that could be used to collect biological and touch DNA evidence. In this video, we are going to show you the processing of some biological evidence and touch evidence by using one of those devices. Keep in mind that the evidence collection process is not static, and that prior to this collection process, the evidence was identified by our crime scene commander in his initial survey of the crime scene, which included an interview with the first responders and verifying that when they arrived at the scene of the crime, they did not observe anyone except the victim's body, and they did not enter into this bathroom. Also, prior to this procedure, the crime scene commander took a full narrative, including details of where this evidence is located, what its condition was at the time when he first saw it, and also a full set of crime scene photographs were taken with and without scales to document this evidence. Now we're going to proceed into the crime scene to collect that evidence. Also, prior to this collection, a filter transfer method was used, taking a representative sample from the biological evidence for a presumptive identification using phenolphthalein, and we had a positive result. So we will proceed now into the crime scene.
Note that this is a portion that we will use more than one individual in the crime scene evaluation. I'm going to process the evidence. Once I process, I will pass it out of the evidence area to be sealed and tagged and marked for evidence past this period of time in order to protect the integrity of the evidence and prevent possible contamination. Also, you'll note that I am in a full personal protective equipment outfit, including full boot covers, and I've double gloved under this circumstances. This is for two purposes. One, because there is so much biological evidence related to this crime scene, there's a possibility of that evidence contaminating me, so I'm taking protection to keep from being exposed to the pathogens that might be associated with that type of evidence. And also, I do not want to contaminate at all. So when I go into the processing area to avoid all possible contamination, I will don my mask at that time. After reviewing the possible evidence in this area of the crime scene and noting the crime scene commander's log and narrative, I've observed the three possible points of biological evidence that I want to collect. Each one will be collected separately so that we will not contaminate them, not knowing whether this belongs to one or multiple individuals. The device I have selected for collection is the contained device, which has the cotton tip head. After I collect this device, I'll be able to put it back into the container, in which I'll pass it on to my colleague. He will tag it for evidence, putting a security seal around it, and it'll be processed outside the collection area. In this situation, I've observed the blood to be coagulated. I don't believe I'll be able to collect anything with a dry rub. So what I'm going to do is that my colleague will drop a few drops of the sterile water onto my cotton top, and I'll come to the first area that we're going to collect the evidence from, and I'll rub that area with this swab to ensure that I've collected a representative sample of this point. And you'll notice that I'm able to get a very good collection, of which I'm going to put back into the container. I'm now passing this on to my colleague, who will put an evidence security tag on it and mark it for evidence outside the crime scene. Because I have determined that there's three possible sources, at this time, I'm going to, since I've double gloved, I'm going to change gloves outside the area. I have re-gloved, and now I'm ready to collect the second biological evidence site that I see. Again, I'm given a fresh collection device. We will saturate this device with some distilled water, a couple of drops, and I will come to the second site, and I'm going to collect evidence at that site. Getting a full representative sample of the material. Again, I will place it in the device, pass it to my colleague, who will secure it and mark it for as our second collection site. Again, because I've now collected that I have a possibility of having contamination, I'll re-glove again. Now I'm re-gloved, and I'm going to collect the third site that appears to be a blood stain and maybe some fingerprints that are not detailed enough for comparison. So we're going to take another unit. Again, he will drop a few drops of the distilled water on my unit. I'll come to this area, and I will use it to collect evidence from this site. You notice that I'm swabbing the whole length of it, because not only do I have biological evidence on here, I possibly have some probability of maybe some touch evidence because of the fingers touching this material. I've collected that. I'm putting it in my device, again, passing it on to the colleague for security tagging, marking it as evidence, So we have collected the 3 sites that we believe are possible areas for biological evidence for further serological evaluation and DNA analysis. Although it wasn't noted on the crime scene commander's list of possible evidence in the bathroom, we have made the assumption that whoever left the blood in the sink had to enter and leave the bathroom. Therefore, the possibility of touch DNA maybe being on this door area, especially at this shield, opens the door for us to process it. So we're making the decision that we will both dry and wet swab both the push pad coming in and the door handle coming out as possible sources of touch DNA. In this situation, again, my colleagues give me a unit, and what I'm going to do with this one is first I'm going to do a dry swab of the area. And so I'm going to just come out and swab the whole plate hoping to get as much of the trace DNA, touch DNA, on my swab using the dry method first. And I'm doing it in and around the door area surrounding that plate. Again, I will enclose that in my unit and turn it back to my colleague, who will tag it with a security tag and mark it as evidence in that situation. Now, we will do the same thing using a wet swab. I asked him to put several drops of the distilled water on the unit, and I will come back the same way and wet swab this area, trying to be as careful as I can to cover the entire area, rotating the tip of my swab to get as much exposure as I can, realizing in a touch DNA situation it only takes maybe 10 or 12 cells to provide a DNA profile. Again, I'm going in and around the area of the door. I'll give this back to him, and we will process the handle on the other side of the door exactly the same way. In that situation, I've also changed my gloves for that side in processing.
As noted during the collection process, it is essential to record the location and condition of all the evidence collected. Effective methods include the use of photographs and placards to document the location of each item, developing a detailed documentation that describes the items, their location, the location and name of the person who collected the evidence, and the date. We should also make a sketch of the scene that includes distances and a descriptive legend. Now let's review the best practices for packaging and documenting biological evidence. Again, we can see that common sense is the theory behind this practice. We know that packaging our evidence needs to be sealed to protect from loss, cross-transfer, contamination, and other changes. We need to seal the package in a manner that opening it will cause obvious damage and alter the package that has been sealed. Items of evidence for we should pack items for safety using boxes and breathing material, marking items with and informing the lab of biohazards that may be present. We should package unloaded firearms in clean, unused boxes and submit them for biological analysis. Ensure that your swabs and evidence are dry. Mark each package with a detailed description that includes the item, the location, the collection, the name of the person who involved, and the date. Integrity seals must be marked to identify the person making the seal. We can mark through the seal with our name and initials. We must keep documentation of all packaging, including the markings, seals, tags, labels used by all agencies involved. This may be necessary to prove that we have not only secured the packaging or secured the evidence at the crime scene, but the chain of custody has been maintained all the way through the process. Once you have collected the biological evidence, you will be confronted with the storage and transportation of that evidence to the lab. Some recommendations include avoid direct sunlight in hot and humid areas. Obviously, the trunk of your car and its back seat are not good storage places. Some items containing biological evidence may be too large to be reasonably stored. For items like mattresses, sofas, and carpeting, it may be appropriate to photograph the item, clearly showing the location of the biological evidence, and then cutting and removing that portion containing the biological material for packaging, storing, and testing. In conclusion, I would like to emphasize the mission statement the courts have established for the crime scene investigator, and note that by adhering strictly to the guidelines and standards that the forensic science applies to its analytical capabilities, we can show by our actions that we have done the utmost to protect the integrity of the evidence that may prove guilt or exonerate an innocent suspect. Note that our job is to thoroughly and comprehensively examine, we call it searching, the crime scene in order to identify, record, collect, preserve, and transport those physical items that can establish that a crime has been committed and provide a link between the scene, the victim, and the perpetrator. Thank you for participating in this webinar. We'll now open the session to answering the questions that you have presented. I also have a reference source that you can refer to. As we started out, this information is coming from the forensic science community. And you can go on to nij.gov, dna.gov, or feel free to contact me or any of our technical service people if you have questions or concerns. The question was the 10-to-1 ratio for the cleaning solution of water to bleach, whether that has been challenged in court or not. To my knowledge, it hasn't been, but one of the things I would rely on is that CDC uses a reference, and that's the recommendation they make. So my take is that that is what the scientific community is recommending, and I think it could be supported with a great deal of support from the community. No, actually, that court mission statement was taken from a variety of cases in which the first sentence that you see, it was to thoroughly and comprehensively examine. What the court was looking at is how can you prove that you thoroughly and comprehensively examined and that the evidence that you were presenting was probative. And that's the whole idea that when we look going back to these things, the reason we do certain things, the reason we double glove, the reason we wear protective equipment is to make sure that we prove that we have thoroughly and comprehensively examined and looked for all the evidence and have taken all the possible steps to protect that evidence from the time we found it at the crime scene until it's presented into the court. We're having some conversation between ourselves because, first of all, if you look at the DNA or the European procedure, they test DNA first and then they go after the fingerprint evidence. I think that's a pretty sound process that is going on, and that I would take as carefully as I could. I would collect samples for DNA that wasn't, keeping in mind I don't want to destroy evidence that I can see that may have ridge details. So I think that I just have to use good sense, and I would go after the DNA evidence first, and then I would process the rest of it for details for ridge detail. The question is taking DNA exemplary samples, and especially when we're dealing with a bathroom. And obviously it has to deal with whether is this a home where we have a relatively few number of people who may be exposed or be able to be in the bathroom, or we were in a public setting where a lot of people have come in. What we're really concerned with is what the people we do know, and that's why we want to limit the number of people there and take exemplars from the people we know have been there. Naturally, as we have done in the past for elimination prints from members of the household, we would proceed to also take elimination prints from the members of this household. In this scenario, you notice that we took the time and effort to talk to the first responders to try to see if they had any information about who was there. We've been in situations where a crime is committed in a house and a neighbor has been in and out of the house prior to the time the CSI people get there, and if that person is not identified somewhere in the log or in the narrative, it'd be possible that that person could leave their DNA at the crime, and we wouldn't have that recorded. Well, we went through that process to go, and you saw when we recommended the types of evidence, probably by a very large majority, the common DNA swab or common swab that is used is what we call the therapeutic swab. It's the less expensive one, and generally the one with the wood handle and the cotton tip. Naturally, if you take the precautions, you're careful, you air dry it and avoid all possible means of contamination, this can work very effectively. And it has worked very effectively and is used effectively every day. And we appreciate your concern about costs because every department in the United States is faced with budgetary restrictions. The question is, what would be a basic DNA kit? And in a basic DNA kit, we certainly would have to have gloves. And I have to say, even though we sell gloves, I would recommend a number of gloves there. Having one set of gloves is not going to be sufficient for it. And having a reasonable amount of swabs, whatever one that you're going to obtain. You know, we're in a world now that we're able to produce DNA-free swabs. Is that what the courts are going to demand in the future? It may very well be. But certainly, and we would want some distilled water in there so we could process, and some containers to put the swabs in. So, and some evidence seals and tape so that we can secure that evidence. So I would say that if we had a kit that contained sufficient number of gloves, swabs, sterile water, packaging material, and seals and evidence tape, that would be sufficient. All right, the question is, why do we collect a dry and a wet sample? Well, we collect a dry sample first because we might be dealing with minute amounts of cellular material. And the cotton tip being dry may pick those up. And that's what we're looking for. If we went with the wet first, we may actually wash those away or destroy them in the process. So we're using precaution to make sure that we can get the maximum amount. So using the dry cotton head at first, the cells may adhere to that, and we pick those up, and then we're coming back with the wet swab to make sure we picked up anything that we've missed on the first time around. Yes, and also we're careful to make a statement that we've labeled in our descriptive information in packaging which swab was our dry swab and which swab was the wet swab. Well, the question was how to determine what to sample and where. I think the answer comes in looking at the object that we're dealing with. Obviously, we can't swab an entire room, so we're looking for areas where people would come in common contact with an area. For example, when we decided to swab the push plate and the handle of the bathroom, that's where we expect people to put their hands. We naturally do that in and around that area. When we're dealing with clothing, we mentioned in somewhere that if someone is wearing a shirt, the areas that come in close proximity to the skin and the sloughing of epithelial cells would be the shirt collar and also the cuffs. One of the things that we've noted from examining prior cases is that sometimes in an assault situation where the perpetrator has grabbed the victim by the front of their clothing in and around the shirt or the button areas or in and around the collar areas is also an excellent place to look for epithelial cells. We had another situation where a mask was involved. Naturally, the opening around the mouth and the nose where saliva would be involved, or nasal mucus, those are all potential areas where we should be looking for DNA sources. The question is, in considering blood, how to determine when and where to swab. To me, blood is one of the few biological substances that kind of waves its hand at you. In a crime scene, the theory is if it looks like a duck and quacks like a duck and walks like a duck, consider it a duck. In the same way with a stain, although it may not have the same presence that we see fresh blood in, if it's associated with the other facts of the crime, we're always going to swab that blood stain and get a representative amount from that. One of the things we have to be careful about are the number of stains involved. We cannot assume that just because we see one stain, that is the exact same stain from the same depositor. We can have a situation where there be multiple perpetrators and their blood could be different. So we have to collect every blood stain that we see at the crime. The question was that the comment was that we should have a full personal protective equipment available in that DNA collection kit. I cannot agree more heartily than with that suggestion. Rarely will people take the effort to do that, but the possibility of contamination, and especially considering the fact that we're using touch DNA and analysis now, for your self-protection and protection of the evidence that we're collecting, we should always be ready to be able to don full personal protective equipment, including head covers, shoe covers, gloves, and a mask. The question is that if luminol has been used to look for hidden blood, has the use of the luminol contaminated the stain, and will it prevent further DNA analysis? There's sufficient literature in the resource materials that identifies that luminol does not destroy DNA, the possibility of obtaining DNA profile, either in the high molecular or low molecular value of DNA. However, that doesn't mean that by using luminol and using the water that is used as a solvent with it, that you may dilute the material to such an extent that you may not have a suitable amount for a DNA profile. That poses the question that the last thing we would do is use luminol. That is a screening device that is used after every observation has been made at the crime scene to do it. And if we did use it, we would use it in as small amount as we possibly could. The question is about submitting control swabs when we process. The best answer is, obviously, that a control swab should be presented with every evidence collection taken. If we're using a standard lot of swabs, we may be able to present that swabs that haven't been used from that lot, and the lab will be happy with that. But my rule of thumb is that there should be a control with every positive. The question is, can we comment on luminol versus Bluestar versus Limousin? All those compounds are luminol type compounds. The major difference is that in the formulation in Bluestar and Limousin, they are using hydrogen peroxide as a catalyst. In the standard luminol type compound, we're using sodium perborate as the catalyst. The sodium perborate is not as hydrophobic, therefore it has a tendency to have a longer unmixed shelf life, but it does produce a lighter luminescence. whereas the peroxide in the Bluestar and the Limousin produces a brighter luminescence. In all three cases, you will see luminescence. If you need to have as bright a luminescence as you possibly can, then I would suggest that you select Bluestar or Limousin. I don't believe that one is much more effective than the other. It's a personal preference in that process. The question was posed by Michael Lafort. It was a comment by Michael Lafort, and Mike, thank you for your comment. When we do use luminol and we start processing, as soon as anything is generated, we stop and tag at that time. For example, let's say we were processing a carpet and we found a luminescence on top. There's a good probability that that blood may have gone under. There's no need for us to continue processing at that time after we've tagged that. So we would suggest that you never overprocess with luminol just for the sake of seeing the glow. It should be used to identify where biological evidence may be and be tagged and collect that for confirmatory analysis. The question is, if we use small particle reagents to test for wet fingerprints or fingerprints on a wet surface, and we're not able to obtain fingerprinting, would there be enough residue evidence to produce a DNA profile? That's a very good question, and the only way that we're able to be able to prove that one way or the other is to go ahead and collect it. If I thought there was sufficient material there to use the small particle reagent, I would then naturally swab first, trying to collect the DNA and then proceed on. So in that situation, the only way is the proof of the pudding is going to be in the DNA analysis. But remember that as few as 6 to 10 cells can be sufficient, so I wouldn't pass up that opportunity. The question is, will we continue to do webinars and other topic areas? And the answer is absolutely. And we would appreciate your input to the type of topic areas that you would like us to work on. We are working our way down the list, and we're hopefully going to be able to generate webinars. And understand these webinars are going to become interactive relationships with the end users. We have some answers, we have some questions, and we would like to pose those to the community and get a dialogue back and forth with it. So send your cards and letters and make your recommendations, because that's what we'll work on. I want to thank everyone for taking the opportunity to be with us. I hope that you found this webinar informative. The process is what we want to continue to do, and with your cooperation and input, we will try to bring to you the most pertinent information that we can coming from the forensic science community. Thank you and have a good day.